Hello, everyone, and welcome to this audio podcast. As part of this audio podcast as part of a Storm and Squala. This audio podcast as part of a Newspart interlude. This audio podcast as part of a Newspart interlude, and this audio podcast as part of a Storm Robotics production. In this audio podcast, or through this audio podcast, a sort of a look at the underlying aspects in regard to chemokine receptors, and leukocyte trafficking in regard to acute allograft rejection, or AAR, in regard to leukocyte trafficking in acute allograft rejection, or AAR, and in regard to renal transplantation. So in renal transplantation, the occurrence of one or more episodes of acute allograft rejection is a major determinant of allograft prognosis. Most episodes are episodes of acute allograft rejection is caused by cell-mediated processes and require the infiltration of alloactivated T cells into the engrafted organ. Over 40 inducible chemokines and 20 inducible chemokine receptors have been identified. Have been have been in have been highlighted and identified in amongst sort of a literature of works in amongst sort of retrospective works and in vitro investigations on responses of activated T cells to inflammatory chemokines suggests substantial redundancy, a sort of a substantial aspect of redundancy in this system with multiple chemokines produced at tissue sites binding to multiple receptors on circulating leukocytes. Chemokines binding to receptors on leukocytes mediate the sort of the behind the scenes plays between antigen presenting cells and host T cells in lymphoid tissues and the actual here and now to propagate an immune response that acutely destroys an allograft, and seems apparent where inflammatory responses where inflammatory responses are seen sort of from a clinically important perspective as where chronic as where chronic rejection occurs. The biological actions of chemokines are nomenclature used to describe these receptors. The receptors is based on the class of chemokine ligands and that interact with receptors. For example, CC, CXC, and CX3C receptors. Many chemokines bind to several different receptors, and the differential expression of the receptors by distinct leukocyte subsets is an important component of the specificity of chemokine action. The complexity and the apparent redundancy of the system is thought to provide a high sense of effectiveness and flexibility in vivo. Investigations with knockout mice or murine laboratory models, especially when teamed with the use of wild-type murine laboratory models of blocking monoclonal antibodies to the corresponding receptors, have provided the first hard data as to what all complexity of chemokine biology might mean to highly practical in vivo world of and in around transplantation. Investigations inform that have informed that expression of multiple chemokine receptors allow a navigation by leukocytes in a systemic fashion through arrays of multiple chemokines. So different chemokine receptor pairs may be necessary for activation, transmigration, and retention accordingly. And there have now been investigations on the expression of several chemokines and chemokine receptors in human renal transplantation that show increased expression in acute allograft rejection, or AAR. Several important points have emerged in amongst the literature from in vivo based mechanistic investigations. First, targeting of a single chemokine is typically ineffective in prolonging allograft prognoses. An example of this general principle or aspect is the lack of efficacy in targeting of macrophage inflammatory protein 1 alpha, whereas targeting of receptors CCR1 and CCR5 is of therapeutic significance. Second, chemokine receptors differ in importance as targets in allo responses. Thus, chemokine receptors such as CCR1 and CCR2 primarily promote macrophage recruitment to an allograft, such that targeting in knockout murine laboratory models has only a modest effect on allograft prognoses. Third, the effects of containment of immunosuppression, for example, can modulate some of the rather black and white outcomes of chemokine receptors targeting in otherwise untreated murine laboratory models. Thus, use of a subtherapeutic regimen of immunosuppression has shown a sort of a synergistic advantage with targeting of chemokine receptor, by which itself has only a weak or negligible advantage. In the case of chemokine receptors, with the most impressive experimental data, experimental data historically to its role in acute allograft rejection expression is of CXCR3 and its ligands which closely related to, which are closely associated with and around the bases in around acute allograft rejection. Data, for example, from around 163 renal allografted recipients concerning the extent to which chemokine and chemokine receptor polymorphisms can affect the incidence of acute allograft rejection to the extent to which a chemokine and chemokine receptor polymorphisms can affect the incidence of acute renal allograft rejection, or AAR. The various stimulatory and inhibitory pathways involved in these activation cascades are and have provided insight into the design of potential pharmacotherapies. That draws this audio podcast to a close. This audio podcast as part of a Newspart Interlude, this audio podcast as part of a Ztorm and Squala, and this audio podcast as part of a Ztorm Robotics production.